for instance like glycine or iodophenoalanine. And in this slide you can see, out of a review by Venture Use Laboratory, just some of the many modified amino acids that can be charged. Some of the individual variants charge over 12 non-canonical amino acids. The cognate amino acid is pile, and the most inexpensive is glycine. So many of these amino acids are quite expensive, and our lab has been the only lab that actually worked with pure pyrrolysine, for which there is a very difficult synthetic procedure. And what I would like to see is that pyrrolysine and pylorus show the same kinetic parameters that the other amino acid tRNA synthetases show. It's very, very specific. But the pyrrolysine tRNA synthetase that you see, that everybody uses, has already 4 amino acid changes in it. And this then leads to the polyspecificity of the enzyme. The overview of pylorus enzymes comes from an analysis of 155 organisms that have 1 copy of pylorus and 3 organisms with 2. But the domain organization is very interesting. There is an N-terminal domain that is essential for activity, and also a C-terminal domain. And there are many organisms that have the N-terminal and the C-terminal domain separately encoded and not linked. The original discovery in methanosarcina showed the N and C-terminal domain linked by a variable size linker up to 180 amino acids. And then the big surprise was a few years ago when it was shown that there is a different class of organisms that have the delta pylorus N, where the C-terminal domain is doing everything. In terms of structure, we showed in 2009 that bacterium Desulfitobacterium hafniense is co-crystallized with the tRNA pile, but this is only a fraction of the tRNA because the N-terminal domain is exceedingly insoluble. So we showed that this is a different kind of complex from all other amino acid tRNA synthetases, where the enzyme is actually binding the inside of the L of the tRNA. And when Tateki Suzuki did the structure of the N-terminal domain, that very insoluble protein, we actually realized why it was insoluble, because it actually has a zinc binding site, and no one knew that there was zinc in there, and this was very, very insoluble. So this is Tateki's structure of the full-length enzyme. So the N-terminal domain is shown here. The C-terminal domain is shown here. And basically, this stippled path here is where the linker holds both parts of the enzyme together. So pylorus has the largest interface of all amino acid tRNA synthetase tRNA complexes. And as you can see here, pylorus does not recognize the anticodon. This, of course, is a blessing for all synthetic biologists, because pylorus then can be used to recode any codon that you like. And lastly, the discrimination against the canonical tRNAs is based on their larger variable arm. I'll show you in the subsequent slides that tRNA pile has only an exceedingly small extra arm, and the canonical tRNAs have one or two bases more, and this is the basis for this combination, as Tateki showed. I now would like to tell you something about methanohalorcheum thermophilum, HMET1. So this is a story of tRNA recognition and the evolution of pyrrolysine. This organism is an exceedingly halophilic, methyl-reducing anaerobic archaeon. It grows at higher temperature at about 86% saturated salt. It's a deep, very, very deep branching uric archaeon. A complete genome sequence is known. However, of course, no usual biochemistry can be done because they're only halophilic proteins. So, to make it work, we had to go and use a halophile model organism, and this was Halophyrax volcanae. So everything was done by really taking the HMET genes, putting them into Halophyrax, expressing them as was shown in this NER paper, where you can read the experimental details. And the story then evolved like this. HMET has two different tRNA pile species. It is the only organism we found that has two different piles. This is pile T, this arrow here, and this is another one. So they are in two different strains. In each case, they are next to pile S, it's the pyrrolysine synthetase. And this is the number one tRNA pile and number two tRNA pile that, among a few other bases, differ in the discriminator base. tRNA pile 1 has a G, as most other tRNA piles, while a discriminator base A is found in tRNA pile 2. And this is this extra arm, which is very, very small. So this is the discrimination between tRNA pile, bipolar and the canonical tRNA. So you can see this. The HMET2 has two polarized enzymes, as we just showed. So there are 13 delta polarized enzymes. And when you do a phylogenetic tree, you can see this. And this is polarized 1, polarized 2, as you saw. And back to this. Here you look at polaris and polaris. And when you look, then you can find there is actually a difference in the important motif 2 loop that is for recognition of tRNA, where polaris 1 has a normal size loop, polaris 2 has one amino acid missing. And there's one other organism that is not completely sequenced, where it's the same, that polaris 2 in MSBL1. One amino acid is missing, and the missing polaris 2 with the missing amino acid recognizes the A discriminator, while all the others recognize the G discriminator. So polaris 2 has one amino acid deleted from motif 2 loop. Archaeal, and this is phenylalanine tRNA, which is, we showed long ago, is a precursor of tRNA pile. And there are a number of them that have a U in the discriminator position, others have an A, and the rest have a G. So of the 105 organisms, G is really the major discriminator. Only 2 organisms that you saw have tRNA-pyle with an A, and 3 organisms with U. So we asked Kuhnen, can we find out, is tRNA-pyle 1 or tRNA-pyle 2 older, ancestral? And it turns out that tRNA-pyle 2, this pair, is the ancestral one and got lost. This is not surprising because most discriminator bases in nature have adenosine. And therefore, to get rid of that competition, very likely this is why this tRNA got lost and it's replaced by G, and the same while it might be U. And pyle is a member of the original archaeal genetic code, was lost by many archaea and also transferred to bacteria. And there may be, the existence of pyle system may be linked to methanogenesis. This is a discussion by itself. I'd like to then come to the last point, initiating translation with non-canonical amino acids in living cells. So the goal was to develop an amino acid tRNA synthetase and tRNA pair to initiate translation with non-canonical amino acids, NCAAs.
Professor Sol, can you condense it, make it a bit shorter, the last part?
Sorry?
Can you make it a bit shorter, the last part, as we are running late in the evening here?
I don't understand. Can I make what shorter?
No, can you make it a bit shorter, the last part?
Dieter, I think what the gentleman is saying is that you should truncate it a little bit, the last part.
Okay. So we borrowed the idea from Raj Bhandari and Varshney, and Tom talked about this in his introduction. So 4-methylphenylalanine and tyrosyl tRNA synthetase from Nashi was taken. And with this then, we had a very, very nice paper with, that had the cover of the journal with Umesh Varshney. Thank you, Umesh. And basically, to make it we could show that with UAG and initiate tyrosine initiator tRNA, we can then get the methylphenylalanine incorporated. The second point was, can we modify these to recognize AUA codons? And the answer is yes. You can take the UAG and checking all the initiator tRNAs, codons, UAU can be used. So this is fine. And if you take normal tyrosyl tRNA, it will not go into the first position. So it is a special tRNA. And the last point then is, we can really take UAG and UAA suppression to put eventually 3 distinct bioorthogonal handles into a protein using the system. So we could put in for UAU, for UAG and UAA different amino acids that can be recognized by fluorescent probes. So this came out of the system. And the last point is, I would like to thank really all the people that worked in my lab. 9 months ago, this was my lab. My lab is now only one person strong. So, Christina Chung, Kexin Meng, Danny Chung, Jeffrey Thorpe, Natalie Crime, Oscar Vargas, and Dominic Schuntermann have really helped over the last 2 years. Felix Fu, Litao Guo, and Patrick O'Donoghue were in the lab and they are elsewhere independent. And when I reported, Eugene Koonin at NCBI, Kira Makarova and Yuri Wald have helped, and also for the first paper, Alana Shepard in Berkeley. I have to thank the Department of Energy and especially the National Institute of General Medical Sciences for support. I was very fortunate. Thank you very much for listening.
Thank you very much, Professor Sol. Thank you very much for this wonderful talk. But being a special talk on a special occasion, we will not have any question answer for this talk.
Okay.
Thanks very much again.
Yours, pleasure.
Yeah.
Thank you very much.
Yeah. So I think we shall proceed with the event all of you here are waiting for. But you have already heard from Tom about the protagonist of BTMO 2023. Whatever I am going to say now, because it would be superfluous and it will not be anywhere near, it will not be on par with what Tom has already introduced about Umesh. Nevertheless, being privileged as chair of this session, I would like to add few things which I think will be useful to the student community of our own institute as well as the students coming from elsewhere. So flashback, going back to 60s and 80s, 60 to 80s, MCB department pioneered work on mycobacteria, early molecular biology, intermediary metabolism, phage biology, and so on and so forth. Come 90s, those scientists had retired or changed their area, and to strengthen the mycobacteria, 4 young faculties were recruited, suggesting, they all came with the different work expertise, suggesting that they could start working on mycobacteria. I think only one of them had prior exposure to mycobacteria. The 4th one selected to work in not exactly microbiome cell biology. It's a center for genetic engineering with the control from microbiome cell biology. The 4th one recruited happened to do PhD in University of Calgary, short postdoctoral stint in University of Calgary, and a longer stint of postdoctoral research at MIT. And Tom has already said what work was initiated and done in MIT by Umesh. I must just add here, the department had already recruited someone who had indulged a little bit on transcription, and Umesh coming in there was like a fitting, because he is, as I said, there was work on translation also early years. That work had stopped in the department. And coming from protein synthesis lab, Umesh joining the department or Center for Genetic Engineering, which soon got merged after a few years with the MCB, was like a perfect fit. What happened then is a coupling of transcription and translation as it happens in a bacterial cell. Transcription has been terminated already, but translation is going on. And what you're going to hear now is the different aspects where Umesh has an immense contribution, whether it's the initiation of translation or elongation and termination, and the translational fidelity, which was not addressed for a long time, and many, many other aspects. But I must add here, before I end and leave the floor to him, that he speaks of science, breathes of science, and lives with science. But I must reveal he has two passions. And I have gone around with him to various scientific sessions, and that passions are DNA repair and translation. I really didn't know which, and these two indulgences you can see in different forums. So I was thinking that he will talk about both of his work, but now today from his talk, you know what is his first love. Now let us welcome the man of the hour, man of the event, Professor Varshney.
It's on. Thank you. Thank you, Nagraj, for that generous introduction. Thank you, Tom, and thank you, Dieter. I will have more to say about Tom a little later, but Nagraj has been a wonderful person. I've worked along with him since '91. We've had joint a lab meeting since '91, and it's just been, a joy to work in the same place. In some sense, Nagraj is actually a role model for me. The kind of multitasking that he can do, I would be really happy if I can do even half of those. So thank you, Nagraj. So I'm going to be talking about the translation part today. About a week ago, I had talked about DNA repair, so I thought I can just take the liberty of talking about the work on translation today. And what I would dwell on is really this particular pathway where we have spent a lot of time. And what you're seeing here is a translation initiation in bacteria where all of these components come together, 50S subunit joints, and then makes the ribosome ready for elongation. One of the things that right in the beginning we wondered was that is there a way to probe this pathway? And the way to do that we thought we can do is we can make mutations in the tRNA such that the tRNA does not go through this pathway in a regular way. But if we force the translation system that it can go through this pathway, we will learn something about how this tRNA interacts with various factors here, or what are the other things in the cell that actually help the tRNA, initiate a tRNA translate protein synthesis. And to do that, I need to introduce the molecule itself. And in E. coli, there are 4 genes for initiator tRNAs. 3 of them are together in one locus, and the 4th one is slightly away, alone. If we look at the cloverleaf structure of the initiator tRNA, there are 2 very distinctive features in this. One in the acceptor stem region, which is important for formulation of this tRNA, and the property of formulation is unique to bacteria. It does not occur in eukaryotes or archaea. The second region is in the anticodon stem, where the initiator tRNA is very, very unique in having 3 consecutive GC base pair, and these GC base pairs are highly conserved in all domains of life. And this is really something which we got our attention got drawn to. So yet another thing I need to introduce here is a part of this slide Tom has already explained. And so the idea was that if we make a change in the anticodon of the initiator tRNA such that it will recognize the termination codon, and the termination codon which has been occurring here in the place of start codon, the tRNA recognizes that UAG as a start codon, and in the reporter gene, which is chloramphenicol acetyltransferase, it will make chloramphenicol acetyltransferase and will confer chloramphenicol resistance to the cell. So what we wanted to do was mutate this tRNA in such a way that the amino acylation and formulation properties are not affected, but its initiation function is affected. And that region in the tRNA was the 3 GC base pairs in the anticodon stem. So we changed those sequence to the one that is found in the elongated species of the methionine tRNA, and then asked a question whether this tRNA will function in initiation pathway. So we use the same assay system, and here you can't see somehow the CUA anticodon is there. So the tRNA is amino acylated well, formulated well, but it does not translate the reporter mRNA, and therefore the cells are chloramphenicol sensitive. So this was a perfect molecule for us to then do genetics, and this work, which was a lot of work actually, was done by a postdoc in the lab, Dinesh, and what he did was he subjected E. coli to mutagenesis. Now in this mutagenesis, his idea was to look for chloramphenicol resistant cells, and chloramphenicol resistance can arise in so many different ways, but his task really was to select for the mutants which would require this mutant tRNA for initiation, and the initiation would occur only from this UAG initiation codon. So this screening, he spent a lot of time, and he isolated many strains in doing so, and these we call as suppressors. These numbers are just arising from the Connolly number, plate number, and so on and so forth. The numbers themselves don't have any meaning, but they served as nomenclature for us. So using those strains, we've done quite a bit of work, and the 2 things that I can tell you based on that work are that the formulation of the tRNA is very crucial for the initial targeting of the tRNA onto the 30S subunit. Its contribution in the subsequent steps of initiation is not very much. But if we look at the 3 GC base pairs, it has very important role not only just in targeting of the tRNA onto the 30S, but also to take the tRNA in the subsequent steps of initiation and making a 70S initiation complex which is ready for elongation. So this 3 GC base pairs are very, very crucial. That work has been published quite some time ago. So now how these 3 GC base pairs are doing all of these activities. Now in this famous paper from Wanke's lab, what has been observed is that if you look at the initiator tRNA, these 3 GC base pairs, they are actually interacting with 2 of these nucleotides, G1338, A1339 in the head region of the 30S subunit, and they make the A minor interactions. Now some of these interactions are suboptimal, and this actually happens, as you will see a little later, because it's a technical requirement that the mRNA binds tightly to the ribosome. So there is actually extended Schneider-Gando sequence that is used in these crystal experiments. So what is happening here is with the extended Schneider-Gando sequence really is that if you look at this suppressor strain that we characterize, this is the normal way that the cat messenger RNA is interacting with the 16S rRNA. There are 6 base pairs that it forms. In the B21 suppressor, what happens is that there is a mutation in the ribosomal RNA from the C to U, and that then allows 8 base pairs. And if you recall, in that crystal structure also, the messenger RNA is interacting with ribosomal RNA with 8 base pairs. So what it must be doing to allow initiation with a tRNA which otherwise would not initiate, it is actually lowering the accuracy of initiation. And so no surprise that there was some of the context that was suboptimal. But this really served a major purpose for us. We could then show that even if we make this change in the messenger RNA so that 8 base pairs can be formed, we can see that there is huge amount of initiation that is coming in this system now, even though there is no change in the ribosomal RNA. So based on many of these ideas, we also then showed that the initiator tRNA and the 3GC base pairs are also important in the ultimate steps of processing of 16S rRNA. And the way it is happening is that in the pioneering round of ribosome biogenesis or the initiation complex formation, once the 70S complex is formed, the ribonucleases can come and chew up the extra sequences that are present on the 16S rRNA at the 5' end and the 3' end, and that can then mature the ribosome for subsequent rounds of translation as well as this first round of translation. Now, something related to the ribosome biogenesis came up in yet another suppressor strain, which is A18, and Kuldeep worked on it, and this turned out to be a mutation in RLUD protein, and the mutation changed glutamate 265 to lysine 265. Now, if you look at this strain, the steady state levels of RLUD remain same compared to the wild type strain. So they don't change. And if we look at the pseudouridine activity, biochemical activity, that also remains completely unchanged. Now, if we look at the literature, what we find is that already there was a phenomena characterized for RLUD mutation, where if they take the catalytically dead mutant of RLUD, it would not do the pseudouridine formation, but it would rescue one of the strains of E. coli which had cold sensitivity. Now, Janos talked about many other modifications in ribosomal RNA yesterday, where also the phenomena was that they affected ribosome biogenesis. Now, yet another observation that allowed us to then follow the ribosome biogenesis was the work done by Eric Brown, and what he showed in his genetic analysis was that this RSGA, which is coming in the same pathways of ribosome biogenesis, had genetic interactions with RLUD, RBFA, and ERA1. So this really then suggested that the RLUD mutation, which is not affecting the biochemical activity or the stability of this enzyme, must be involved in ribosome biogenesis. And if we look at the pathway that we were probing, if you look at the RBFA removal from the 30S, this is actually carried out by RSGA. Recently, it was shown that initiation factor 3 will also do this job. And so in both of these cases, RBFA is released and then our ribosome can mature. So the question that we were asking is RLUD also involved in this pathway? So we did a lot of genetic analysis because you can make predictions from these factors that are involved, and you can make predictions whether the initiation with the mutant tRNA that we are dealing with, the 3GC mutant tRNA, whether the initiation will go up or down, and all of those genetic predictions were met here. So then that then allowed us to ask the question whether RLUD is actually removing RBFA from the ribosome. And if you look at just this slide here, this is the fraction of 30S subunit. This is the situation in the wild type strain. And if you make a mutation in RLUD in the genome, you find that there is more RBFA on the ribosome, which means the ribosome actually are not completely mature to do the protein synthesis. So then this observation allows us to say that RLUD is also involved at this pathway. Now, we do not know how this is functioning, but a prediction would be that RLUD is actually binding to helix 69. It actually does modification of 3 residues in helix 69. This helix 69 will dock on the 30S right at the place where the RBFA is bound. So, it's quite possible that when the 50S subunit is docking, this the RLUD is somehow involved in removing this RBFA from the ribosome. Now, let me switch to some of the work that we have done where one carbon metabolism turned out to be very crucial for the accuracy of protein synthesis. Now, one of these example comes from the suppressor B2, and what we found in B2 was that the FMT gene, which makes formal transferase protein, there was a mutation at 274 position in the gene where a glutamine was changed to an AMBA codon. Glutamine codon was changed to an AMBA codon, and that if there was a termination at this place, it will truncate the protein by 42 amino acid. Now, based on some of the work that has been done in Tom Rajbhandari lab, we predicted that this mutant would be completely dead, because even if you remove 20 amino acids from the C terminal and the enzyme loses this activity. So, when we checked the activity of this enzyme, there was about 2% activity of FMT in this delta 42 strain. And now one can argue that maybe this is the residual activity, and therefore we looked at whether it is coming from this kind of a strain or there is something else happening. And if you look at it, in this mutant background, actually the full length protein is made. And this protein is made only when this mutant initiator tRNA, where the 3GC base pairs have been mutated and it has CUA anticodon, only then it is produced. If you have vector alone, this protein is not produced. So, it suggests that this little bit of protein that is being produced is what is responsible for this 2% activity. Now, if this activity is real here, we should also be able to tell it with the acid urea gel that Tom Suzuki and Tom Rajbhandari, both of them talked about. And what you will notice here is in this particular instance, so this is the wild type background, and you will see a single band which corresponds to formulated species of the tRNA. In B2 suppressor directly, you will see that there is yet another band which corresponds to the amino acylated form, and that amino acylated form will disappear when you treat with the copper sulfate. You see that deacylated, but the formulated species remains there. And in a strain that where we ourselves generated AMBA 274 mutation in the genome, there also you find that there is partly amino acylated tRNA present. Now, the reason for us to look at this really was that this is an initiator tRNA. Its known function is to take part only at the step of initiation, and to make a full length protein from a mutation at 274 position, which is AMBA codon, this initiator tRNA must participate at the step of elongation. And this is really what I am trying to tell here, that normally this tRNA will be formulated, but if the amount of FMT is much less, there would be some amount of tRNA which does not get formulated, for example here, and perhaps this binds to EFTU, and this can then take part in elongation. And therefore, we should be able to show that this tRNA is binding to EFTU, and this is really what Riaz did. He showed that this particular tRNA in this background will bind to EFTU. This is the control, but in the wild type background, this tRNA will not bind to EFTU. Both of these, in both of these background, the initiator tRNA obviously binds to IF2, the functions in initiation as well as now here in elongation. So, this really then tells that EFTU binding is what is happening here, and that is happening with the partly amino acylated tRNA that is remaining in amino acylated form. Now, another connection to this one carbon metabolism came from these 3 suppressors here, where there was a mutation in folate dehydrogenase. Now, if you look at the one carbon metabolism, folate dehydrogenase is a bifunctional enzyme. converts 5-10-methylene tetrahydrofolate to 5-10-methionyl tetrahydrofolate, and then it goes into the 10-formyl tetrahydrofolate. And so these three suppressor strains had three different alleles of folate dehydrogenase with a partial activity for folate dehydrogenase. And if you look at A48, B22, and A30, Gautam in the beginning worked on the A48, and then these two suppressors were worked on by Shivji and Kuldeep. And if you look at these activities, this is the wild type. You see the formulation activity quite well. But with these three mutant strains, the activity is much less. And the least activity is by C58Y. Now, we know a little bit more about these things, but in the interest of time, I'll just move forward. And what you will see here is, if you use this reporter system, where we're looking at the initiation from UAG codon, all of these suppressor strains, of course, initiate. And this is our control now, where the mutation is in FMT gene, and that also initiates. You will also notice that if you use the system for elongation, where there is EMBA mutation in LEGZ gene, this is the control where we have authentic elongated tRNA, which will suppress this EMBA codon, and you see a lot of LEGZ production. You see LEGZ production even with this particular strain, and you see quite a bit of LEGZ production with the C58Y, which has the least folate dehydrogenase activity. If you do the enzymatic assays, you find that all of these three strains actually initiate as well as elongate. So this really works that, you know, the tRNA is now distributed into two forms, the aminoacylated form and the formulated form. Aminoacylated goes and participates in the elongation step, and the formulated form goes and participates at the initiation step. So now the point here is that the original tRNA in the wild type background was inactive both as initiator as well as elongator. Under the levels of low FMT or N10-formyl tetrahydrofolate, this mutant tRNA, which was inactive altogether, can now serve a dual function. It can participate at elongation and initiation. So the question is, how does it do it? Now, if we look at this, very primitive model here, let's say we have these two tRNAs. Both of these tRNAs are aminoacylated, formulated equally well, but in the wild type background, only this participate and this one doesn't. And that is the reason is because, you know, they both have to bind to the ribosome. In the wild type background, let's say you have 100% formulated of this and 100% formulated this. The tRNA with wild type component of 3GC base pairs has the priority to bind to the ribosome. So all of the ribosomes, 100% of the ribosomes are occupied by the wild type initiator tRNA, and therefore this mutant initiator tRNA does not get an opportunity to go to the ribosome, and therefore it does neither initiation nor elongation. Now, if we imagine a situation that only 70% of each of these tRNAs are formulated, you now have ribosomes which are not completely saturated with the wild type tRNA. So you have some of the ribosomes available to bind to any other tRNA which is also formulated. And that would happen to be the 3GC mutant tRNA. Now, another thing that you can notice here is you also have tRNA which is not formulated. And so this tRNA now becomes available to bind to the EFTU and participate in elongation. So just putting together this cartoon form now, under the conditions of high level of formulation, the wild type tRNA shown here in the green will have the priority to bind to all of the ribosome and carry out high fidelity initiation. Under the levels of low formulation, you will have a situation that you don't have sufficient tRNA which is formulated, indicated by these red squiggles here, and then the ribosomes are available to bind to the mutant tRNA, and therefore there is some amount of low fidelity initiation. Now, you can make a prediction from here that if you actually cut down the amount of this wild type tRNA to half or 1/3, the same situation should happen that the 3GC mutant should initiate. And this work was done by Lassia and Suman. Suman had characterized a set of 3 other suppressors, C5, D4, D27, and in all of those suppressors, what was happening is the promoter for METZW was heavily downregulated. And so the production from this locus was almost nil. And so the amount of the tRNA that we had was much less, and therefore 3GC could initiate. She could mimic the same situation by actually deleting this locus altogether, and showed that this particular strain will now initiate with the 3GC mutant tRNA, as well as a suppressor tRNA, regular suppressor tRNA, sup-E, will also initiate and participate at elongation step. So this work was done, and subsequently Lasia carried out some work, and we also had some collaboration with Janos here. Sorry about that mistake there. This work then showed that there is some connection, or the tRNA abundance really makes a difference in terms of initiation. So what we have shown so far is that B2, A30, A48, B22, where there is downregulation of fold-D, downregulation of FFT, or downregulation of initiator tRNA. All of these things are telling that the divide between the initiation and elongation functions of a tRNA is somewhat blurry. And in fact, if you look at the mitochondria, in metazoan mitochondria, the same tRNA for methionine participates at the step of initiation as well as elongation. This is very much unlike the situation in E. coli, where you have dedicated tRNA for initiation, a dedicated tRNA for elongation. So the question that we asked, can we sustain E. coli on a single tRNA mat? And another question that we asked was, can we do it without tampering with FMT and N10 formal tetrahydrofolate levels that our suppressors did? And to do that then was, basically, if you look at these things, FMT is helping, N10 formal tetrahydrofolate is helping, but the tRNA itself is also involved there. And while working in Tom's lab, I had actually characterized many of these tRNA mutants in the acceptor stem region, where changing this position, this position, this position, or these positions actually made the tRNA very, very compromised for formulation property. And therefore, then Ashwin in the lab decided to take this forward, and his task really was that if you look at these loci for initiator tRNA and elongator tRNA, there are 2 genes for elongator tRNA, and there are 4 genes for initiator tRNA, that he should be able to knock out this, and all of these 6 genes he should be able to delete and ask a question whether the strain now survives on these mutant tRNAs. And this is precisely what he did. He gave a definition of DK, meaning both the initiator tRNA loci are gone, EK, both of the elongator tRNAs are gone, and TK, triple knockout, all of these genes are gone. And he was able to achieve this, and he could actually do the double knockout with almost all of the mutants that he tried, because they are only looking for initiation function. He could do the knockout of elongator tRNAs with these mutants, and he could do deletion of all of the 6 tRNA genes with these 3 mutants. And at 37, there is not a huge problem in growth. With 22, of course, this strain doesn't grow that well, and that is because ribosome biogenesis problems. And basically, if you now look at what is happening here, so these are the elongation function, initiation function, these mutants sustain both activities. And if you now compare, the U1, U3, A70 was the best mutant in the earlier slide. And if you compare these with the human mitochondrial tRNA, you will notice quite a bit of resemblance here. So I think the evolution has really come up with the idea of how to sustain this strain on a single methionine tRNA. So basically then, what was really known, if you look at the tRNA going to the A site, initiator tRNA going to the P site, the idea was that this tRNA will not bind to EFTU because of this mismatch, CA mismatch at the top of the acceptor stem. But what you have seen that this does happen. The knowledge was also that the amino acyl tRNA, which is the elongated tRNA and has very good affinity for EFTU, will not participate at this step of initiation because it will not be recruited on the ribosome. But you have seen that the amino acylated tRNA, irrespective of what is there at the 172 position, will also participate at this step of initiation. So now if you look at the fidelity aspects of the tRNA, essentially what we have in a regular system, that the formulated tRNA will bind to the P site, amino acylated tRNA will bind to the A site. But what you're noticing is that the methionine tRNA can bind to the A site as well. And if you delete the tRNA or you deplete the amount of initiator tRNA, the regular elongated tRNAs can also bind to the P site. The other things that I have not talked about really in much detail here is that 1-carbon metabolism is also producing S-adenosyl methionine, and S-adenosyl methionine is used as a cofactor for many of the methylations in ribosomal RNA, and they also confer fidelity of initiator tRNA binding. So this really then talks about quite a bit of 1-carbon metabolism role in initiation. If you now look at this 1-carbon metabolism pathway, what we wanted to see was whether we can make use of the very, very old antibiotics to do something. Now, trimethoprim has been used very, very conventionally to inhibit bacterial growth, but the resistance arises. Now, what we wanted to do, Kuldeep showed, was that if you actually have fold D mutation, as shown here, at much less concentration of trimethoprim, you can actually kill the bacteria. This is the wild type bacteria growing happily with 1 microgram per ml trimethoprim, but fold D mutants actually are highly susceptible now to trimethoprim. So it's quite possible that then if you have inhibitors of fold D, that the old antibiotics can be revived. Another thing that we have done here is we have actually shown that there is some sort of autoregulatory loop that is established in the FMT mutation. So this is the pathway that we have, amino acylated tRNA getting formulated with the help of FMT and N10-formyl tetrahydrofolate to give rise to formyl amino acyl tRNA. If all of the tRNA is formulated, nothing will bind to EFTU, and you will not be able to suppress this AMBER-274 codon. But if there is a very low level of FMT, very little will get formulated. This can bind to EFTU, participate in elongation, give you FMT, and so there can be some sort of homeostasis that will be established in the cell, that there will be only certain amount of FMT that will be made, and this can actually then be exploited to do regulated protein synthesis. And of course, we can regulate that protein synthesis by FMT levels, N10-formyl tetrahydrofolate, and something I have not shown is by trimethoprim also, because this will also lower the amount of N10-formyl tetrahydrofolate. And Kuldeep has actually shown that by taking trimethoprim, you can make the tRNA participate at both these steps. So the other thing that I will not dwell much, this has been talked by Tom Suzuki this morning, that the intermediates in the 1-carbon metabolism will participate in tRNA modification. They are also important, of course, in modifying the elongation factors, the termination factors, and all of them really. Then basically, 1-carbon metabolism becomes a situation where the protein synthesis can actually be regulated quite well. We think that in the region for methionine as initiating amino acid and formulation of this methionine in bacteria and mitochondria probably serves a regulatory role in terms of the fact that that only when the energy status of cell is very good would you like to initiate. Now, there is one thing that I also want to mention here briefly about the accuracy of initiation. We know quite a lot about abortive transcription initiation. I suspect there is also a step of abortive translation initiation, and that that accuracy here, if let's say wrong tRNA binds to the 70S, that accuracy is maintained by ribosome recycling factor, and Suparna talked about RRF yesterday, so I won't spend much time on that. But RRF works with EFG as well as with IF3. And over we have seen that this RRF can actually disassemble the initiation complex that have been assembled with incorrect tRNA. Now, this has not really been talked about by a lot of people, but as I said that in many of our studies, even when we have not intended to study this thing, it has come up that RRF is important for accuracy of initiation. And we can show by genetics quite nicely that there is genetic interaction between RRF and PTH. PTH is a protein that hydrolyzes any of the peptidyl tRNA that falls off of the ribosome. And to recycle that tRNA, you require PTH. And there is a good connection between RRF and PTH. So I believe that there is another step of accuracy here, which depends on ribosome recycling factor. I want to now very quickly talk about IF3, and the work on IF3 in the lab was started by Shreya. And what IF3 has, it has two domains, N-terminal domain and C-terminal domain. Biochemical properties have shown, or characterization has shown, that a lot of these activities of initiation factor 3 are carried out by C-terminal domain alone. The role of N-terminal domain seems to be only providing additional binding energy of IF3 towards ribosome. So the question that Shreya asked was that if this is doing all of the function that IF3 does, can we sustain bacteria with CTD alone? We delete the NTD and ask whether bacteria will survive. And she was able to do that. She was able to replace the chromosomal initiation factor 3 gene with the full length IF3 as it should. She was also able to sustain E. coli with the mutant of IF3, where 55 amino acids have been deleted N-terminal, and also with another construct where 75 amino acids have been deleted. So this construct has only the C-terminal domain and a bit of a linker. If we delete this linker, the protein is unstable, so we couldn't trim it down further, but this was sufficient to say that we can sustain E. coli with CTD alone. Now, if CTD alone can do the job, why have the NTD? And what she then found was that, yes, NTD is not essential for the survival of bacteria, but it is required for accuracy of initiation codon recognition. And so if you look at it here, the vector in this particular strain where 55 amino acids from the N-terminal are deleted, this is the amount of initiation from AUA codon. If you overproduce the N-terminal domain, this goes down. CTD doesn't do that much. Likewise, if you have AUU start codon, again, overexpression of NTD brings down the initiation. And you can see in ACG. So these are the unusual initiation codons. And if you don't have NTD, there is initiation. But if you have NTD, now the initiation is compromised. So this is required for accuracy of initiation. Now, in the meantime, when we just finished that work, Tanveer, who was at CCMB in Shankar's lab and then went to Venki's lab and came back and joined us as a faculty here. Another student in the lab, wanted to do this work, and what he got help from Tanveer was that there is interaction of the 3 amino acids from N-terminal domain of IF3 with the elbow region of the tRNA. And so what was then done was Rishi from Tanveer's lab actually showed that if you mutate these 3 amino acids, what you will find is that the interaction between the elbow region and the initiation factor 3 will be disrupted. This is done simulations up to 1 microsecond, which is actually quite a lot. But in the case of wild type, the stability is there. What then Jitendra do in the lab was he actually mutated the initiation factor 3 region in the genome, and he inserted a kanamycin locus at the original locus. We have to be a bit careful because there is a promoter for one of the ribosomal proteins in MC, so he was very careful doing that. And then because we were familiar with the CHB locus, because not only with Nagaraj, Mahadevan was also part of our group, and so 3 of us used to have joint lab meeting, and this is Mahadevan's favorite locus. And so we knew that under regular conditions we can do away with this locus, and so Jitendra actually inserted the wild type initiation factor 3 or any of the mutants of the initiation factor 3 at this locus, and he could show that these mutants are produced in the cell, and he could also show if you just focus on this triple mutant here, that initiation with AUU goes up. Likewise, AUU initiation also goes up. He was also able to show that initiation with the 3GC mutant tRNA will also go up, and initiation factor 3 is also involved in inspecting the 3GC region in the initiator tRNA. So then we can summarize this part really basically saying that if you have NTD, there is interaction with the initiator tRNA, and this interaction is very crucial in actually displacing the CTD from where it originally binds and allowing a perfect location of the initiator tRNA in the P site. And when you lose this contact, that just doesn't happen, and everything sort of falls apart. We also got a connection with initiation factor 3 in yet another experiment, and the role of initiation factor 3 in recognition of the 3GC base pair by another experiment that was done by Madhurema. And what Madhurema was doing was, of course, this GC is the wild type initiator tRNA. And of course, when you have the wild type initiator tRNA on a plasmid, you can easily knock out both the loci for initiator tRNA. What she did was she took CG, GC, CG, the first and third base pairs have been inverted here, and this tRNA actually really doesn't initiate that well. In that case, when she wanted to knock out these 2 genes, loci, she couldn't do it. But she could do it if there was also a mutation in initiation factor 3. She could do it, but this strain grew quite slow. So she evolved it in the lab, and so here is the wild type strain, and this is the CG strain. She could get suppressors, suppressor 1 and suppressor 2, and when she sequenced these suppressor 1 and suppressor 2, what she found was that there were mutation in the S12 protein. And so now, she's opened a lot of things here, saying that it's not just initiation factor 3, there seems to be connection with S12 protein also for the accuracy of recognition of the start codon. So let me just summarize here, and what I have shown is that the methionine that is as initiating amino acid has a connection with 1 carbon metabolism. Formulation is important for initial binding of the tRNA to the ribosome, and 3GC base pairs have a lot of function. In fact, I haven't really talked to you much about it except for this accuracy part, but these are all really the jobs that 3GC base pair do. I have also not really talked extensively about the levels of initiator tRNA and its role in initiation accuracy, except for one line that Suman and Lassia did. Now, I will very quickly go through these things, and I want to just sort of let you know that next 5 years I have a lot of work to finish, and methionine as initiating amino acid, and what we want to do is really ask a question whether we can initiate with non-methionine amino acid on a global basis. And so the idea then is that can we initiate with alanine, because the 370 base pair we can change to GU, and that gives a perfect recognition by alanine tRNA synthetase. So also you will notice that in tmRNA, this molecule is amino acylated by alanine. So this work is being done in the lab by Divya, and many of you would have seen her poster yesterday, and she's using these mutants of the initiator tRNA to follow up on this. The other project, of course, that we are doing is looking at the role of formulation, and this work again is being done by Divya, And what we want to ask is that, we know that we can do away with formulation if we produce a lot of initiator tRNA. And there is also evidence that you can initiate directly with 70S. Do we require formulation for 70S, or do we require it only for 30S, or whatever else we have? So now okay, I think I'm running over the time. Yeah. So the other work that is being done is on the initiation factors. Now, we had shown some time ago that mitochondria actually does not have IF1. And so who does the function of IF1? There is an insert of 37 amino acid in IF2, which locates exactly at the same position as IF1 will. And so this 37 amino acid is important for the function of IF1. Now, this IF1 is not known in case of yeast also. And Jitendra is actually looking at which protein might be important, and he seems to have narrowed down on this particular protein which might be doing the job of IF1. Another work that is going on the role of lamotrizine in biogenesis, I'll very quickly go through it. And yet another work, we want to do it in mycobacteria, where we developed a system of down-regulating the initiator tRNA production. And this work is being done in collaboration with Vinay and Nishit Agrawal from THISTI. This work now is being carried forward by Kriti in the lab. There is another work where we want to look at the regulation by interaction of the two tRNAs in the cell, that they can regulate the levels of initiator tRNA. We are also interested in looking at the accuracy of initiation in the T-third ribosomes, where both the 16S and 23S RNA have been joined in the cell. RLUD story is okay, but this suppressor A18 has yet another mutation, which is in RPOA, and we are still trying to figure out what the role of this particular mutation is. Kuldeep thinks that this has connection with transcription-translation coupling, and we like to look at that. Other works that are going on in the lab, and we like to finish up, is looking at the UDG and UDGX proteins, and these 4 people who are in the lab are looking at it. We sort of discovered a protein, UDGX, which makes a covalent complex with the DNA, and why on earth a cell will do that. So we want to look at what the physiological importance of this is. Yet another work that we are doing is MUT1 protein. One of the proteins in mycobacteria, MUT1, has extra domain in this protein which codes for, which actually has the activity of histidine phosphatase. And what Hassan in the lab has shown, that this domain will actually dephosphorylate many of these sensor kinases. So this work we are doing with Deepak Saini in the institute, and at some point I think Hassan will talk about it. My work has been supported by IISC, DST, SERB, DBT, CSIR, ICMR, DBT, IISC. We also have infrastructure support from FIST, and I've been getting this chair professorship money also, and that's been very helpful. I want to very quickly go through the people who have worked in the lab. Now, I have to make a disclaimer here that I haven't been able to put the photographs of all of the people who have worked, and usually one can apply filters to narrow down the list, and you know that filters don't always work. So there is a sort of limited list of people who have worked. So this is the group where people have worked on initiator tRNA structure function, and you will see that even after applying all kinds of filter, this person will figure in every slide. This is the group that has worked on tRNA structure function. 3GC group is this one here. A lot of people who have worked on this. We have translation factors, so RRF, IF3, and many other translation factors. These people have worked. The UDG group, I think many of them are here. These are the people who have worked on UDG. UDG continues. Many other people have worked. Current group really is this one, working on protein synthesis. This group is working on uracil DNA glycosylase. I have a lot of collaborations. In fact, I've enjoyed working with a lot of people in the institute and outside of the institute. And again, I had to apply certain filters here so that I can narrow down the list. And Nagaraja is here. Of course, we have lab meetings with Nagaraja and Mahadevan, but I've also published with Nagaraj. And so Nagaraj is there. Deepshikha we have published with. So all of these people I have published with. And these are also, again, people who I have published with. Other collaborators, so you will see when I again figuring out here. Many of these collaborators actually I have not met. These two people I have not met. So finally, I want to say a few things about the people who have really influenced my life, my science, and everything. And really, I think when I was a master's student at Pandnagar, G.K. Garg really influenced and he motivated to do science. This is really, if he was not there, I don't know where I would be. Lashitu Gedamu, my PhD supervisor, very, very helpful. In fact, I learned a lot of things of setting up the lab. I was his first student from Lashitu Gedamu. And one important thing was that even if I make mistakes, if I make mistakes elsewhere in the university, he'll pick up fights for me. So he really always helped me. Hans van der Sande has been very important in the way that he left me completely alone in the lab. So basically, whatever I did in the lab was my training as independent PI. Tom Rajbhandari, you saw him earlier on, has been really, really a wonderful friend and supervisor. I've learned a lot from Tom Rajbhandari in terms of how to ask questions in science and how to really do science. Tom is also actually a walking encyclopedia. In those days when Google was not there, you can just catch hold of Tom and ask him anything in biochemistry, anything in microbiology, and he will immediately answer that. I always looked forward to his, walking into the lab so that I can just talk to him. Many times that meant that if I was in the middle of my experiment, I will stop the experiment just so that I can talk to him. And then, of course, I have to redo those reactions. Tom doesn't know about all of this, actually. So many times I've repeated the experiment just because I didn't want to lose the opportunity to talk to him. And of course, Govind has always been really a lot of inspiration for me and has been very, very important person that way. This is my group. Last year we went for lab retreat after many long years of COVID. And so this gives an opportunity, and D.N. Rao is right there. He's really the in charge or the director at that place and helpful in arranging this one. I really, as I said, I can't really acknowledge everyone who have really helped me in my tenure at IISC. There are a lot of friends and well-wishers. I've really not mentioned their names. MCB family is really very, very friendly family that we have. I do want to thank my current bosses, Usha and Kumar, to do this symposium. All of you who have traveled long distances to come to this meeting. I want to thank really Sachin, Ravi, Amit, who have really done a lot of hard work. In fact, in this meeting, I've done really nothing. So all of these people and everybody else has really chipped in a lot of hard work. And of course, the students have been completely indispensable. They are the one who have done a lot of work. So a lot of thank you to all of you. I guess Deepshikha has been the total initiator of this whole meeting. And I can't thank her enough, or shouldn't thank her at all, because she put me at this spot here. So thank you all for this, doing this thing. I've also had, the privilege of working in many, many offices at IISC, and I have to say that everyone there has been very, very cooperative. I could just sign the papers many times even without reading them, and they were perfectly fine. So thank you, all of the people in so many different offices that I have held. This is my family, always very, very supportive. Archana really takes care of everything, leaves me completely alone. And she's also responsible for bringing me here, actually. If she did not look for the position in IISc, I would be wherever I was. Our daughter really is very, very independent person, and a very high moment in the life of the parent comes when they are known by the name of their children. And she did that at the age of 5. So it's wonderful, to have the daughter like Ruchi. Ashwin is more like a son rather than son-in-law, and this little guy, Madhav, is always joy to have around. Another one, and thank you. I do want to say a word about this thank you slide. Always is meant to thank the audience, which of course it does mean. But what is also implicit in slide is the Indian Institute of Science. IISc has given me a lot, and there's no way that I can actually thank IISc every which way. If there is something that I have not been able to do, it's not because IISc did not provide. It's more because I did not put my 200% to it. So thank you, IISc, and thank you all of you.
So, after this talk, there will not be any question and answers. There is no question. You can. There is. As a chair, I'm going to overrule any question answer session for this, but I'll with a caveat. The one later on, we have a happy hour soon to follow. That is the time you can indulge in social, scientific, any other questions, and Umesh will be fresh even after a couple of drinks, I can tell you. And so you can go ahead and let us applaud him once more for this wonderful job.
I thank Professor Uttam Rajbhandari and Professor V. Nagaraja for acting as chairs for the session. With this, a day of illuminating talks comes to an end. I request the audience to proceed to Jawahar Visit House lawn for the much-awaited gala dinner. Delegates can assemble in the lobby area to depart for the destination. Bus and cabs will be waiting outside. Students are requested to go by walking. In case of any queries, please contact the travel desk. Delegates staying in MGH, CVH and Shangri-La who would like to avail a cab for their departure to the airport can let us know their details at the travel bench at the earliest. Please treat this as urgent. May all of you have a great night ahead. We will be looking forward to meeting you all again tomorrow morning at 9. Thank you.